Jake, do you think you're at your max of your tether right now, or do you think you
Why are
guys are
we?
what?
Oh, because we need a reset.
Yeah, exactly. I've been waiting for. We've just been on the move.
Yeah, I have to catch up here.
Yeah, so I stopped the ship.
Sats back up.
Hey folks, I was able to join back in. Satellite does keep dropping off, so if I stop in the middle of a sentence, it's probably not because I stopped talking. Apologies for that.
Yeah, why don't you just do a reset just so we're.
All right, I'm gonna do it right here. So you're off your auto. Yeah. Whoops. Wait 1 second. Okay, done.
Can you catch me up? Are we going to stop the ship so we can do a little bit more
Yeah, we
zooms
just
and...
stopped and we have no ship move right now, and we just reset our positioning. So
Okay, so can we pan left and go and look at what we just crossed over, or
We
maybe
need to
zoom
move
out so we
back
can see
then.
what's coming?
yeah,
If you want to go where we were, then we need to move the ship back there.
Well, it's okay. I mean, if we see something coming, I'm fine to check another spot out, but I'd like to be able
yeah, I
to
can
do
go
some
back.
zooms and close-ups if we
Well,
can.
maybe there's more
Oh,
outcrop ahead here, right?
yeah, probably. I mean, this habitat. I mean, in the map, it looks like the exact same environment, and we're following kind of the
That
contour.
would be better.
Okay,
I
so
don't.
let's just keep going, and I'll make sure to
That's
You know,
fine.
stop the
the
ship
problem with
when it.
going back where we were is that we've disturbed it. So
Yeah, and if
Right.
yeah, I'll just try to be quick on
It's just hard to
especially
stopping.
tell
when
you
we
guys
were,
in advance when we want to stop
yeah.
because we don't know. It takes a while to get the ship to stop and things.
Yeah, no,
So
we were just.
you tell me when we can do some close-ups on the next patch that we
Yeah, let me
encounter.
just get in a better position for being able to stop.
Argus was kind of
Great,
leading the way
thanks.
for a little bit, so.
Yeah. All right. So do we have any outcropping up here?
Looks just,
In the sonar, it sort of looks like there's stuff.
yeah, this slight bend.
What's going on with bubble there?
Yeah, and just let me know when you're in a good position.
Yeah, I'm in position again.
Okay, I'll keep going. Is that like a frogfish or something? Oh no, schmutz. It's like looking at clouds sometimes.
This dive seems to be a little boom and bust. We'll hit a big aggregation of animals and then
Looks
maybe
like
a
we're coming into some
lot of
more.
mud. Yeah, it does. I see it's very little current though from the movement of the snow, so I'm curious.
It's really shifting around a lot.
Is that one of the Swiftias that we've been seeing? If so, Meredith
The
would
white one?
love a sample.
Yeah, those red pink corals.
Coral, just in front, just right there.
The red one or the white one?
Red one.
The red one.
Can we zoom in, Dave?
Meredith, how's this for a sample, and what proportion of the organism would you like to sample? Yeah, let's sit here and wait for the visible to clear and get some nice, oh yeah, nice close-ups of this.
Yep.
Beautiful. Pan up just a little bit if you could please get the full effect. Yeah. Oh, great.
So the pink is the skeleton and the orange are the polyps on it?
Yeah, actually with these critters, with octocorals generally, the skeleton is actually many small fragments, and then the central axis. So that their skeleton is partially embedded in the tissue.
Oh, wow.
But this group has a proteinaceous skeleton rather than calcium
Like
carbonate.
Because
So there's a
carbonate
protein called
doesn't do well
gorgonin.
at depth.
Yeah.
Could you widen that shot a little bit, please? I want to get the coral on the top left in the shot. Thank you. Is that the orange cup coral, Balanophylla elegans, by any chance?
You want to zoom in on it?
Yes, please. If so, I would love to get to collect that
Zoom in,
cup
Dave?
coral as well as some of the Swiftia. Meredith said that we can try and just take a piece of the Swiftia, but they may pop off because they seem to pop off pretty easily. Yes, that's beautiful on the cup coral. Can we go any tighter on that?
I think I might need to move in.
Yeah, we're zoomed in
Closer.
all the way right now.
Okay, thank you.
Yeah.
Want to get closer?
Let's do some sampling. Are you close enough to do a little
You want to
sampling of
zoom
that
all the way out
Swiftia?
there, Dave? No.
So
Let me. I gotta
Okay.
move up a little bit anyway. So.
Perfect. Let's move, please.
I don't think these are the target Balanophylla that we're looking for. We're a little bit too deep for that depth range for those species.
Thank you.
Something
Steve.
else. Yeah, cup corals
When I
are
see
just one
those
of those.
solitary corals, makes me wonder. Have the desmophyllum at this depth, but it doesn't look like that.
Want to zoom in again, Dave?
Yeah, cup corals are one of those groups that it's really tough. There's the obvious ones like the desmophyllum, Javania, maybe even Caryophylla sometimes. This looks like a Caryophyllid. That's about as best that I can do using this symmetry.
I love when you say the
That's
obvious
actually
one.
fantastic and far more detailed than I could go, so thank you.
It's really tough, right? Because the tissue blocks all of the structures that are important for this classification. And so oftentimes,
okay, what are
move
we
your mic a little
sampling
bit
now?
closer to your mouth. Yeah, the pink one first.
And where's it gonna go?
Yeah.
If you can get into the starboard box, that would be the best. I think these things don't have a strong hold fast, so the whole thing might pop off.
Okay.
In which case, you're probably going to put it in one of the outboard
Zoom out, Dave.
starboard boxes.
Okay. So when we get ready to put it away, we got to turn off the starboard vertical. So you got to go to relay isolation. And thrusters. And then the starboard vertical command, we'll turn that off. Up on the right, SV command, yeah. So that way we don't blow it out of the box. But we don't need to do that yet.
So while we're setting up to do a little sampling on the Swiftia colony, I wanted to provide a little context as to why I was asking about the Ballenophilia. It is actually part of a graduate student project to look at the skeleton of the cup coral and look at how ocean acidification has
Zoom in a little, Dave.
affected the California current upwelling ecosystem over time. So the orange cup coral will be sampled between here and Southern California
So
to build that
sorry, you want a piece of this
record.
or you want?
I would say go.
Yes, a piece, please, if possible. The whole thing may pop off because it's hard to get a piece, but we'd prefer not to take the whole colony if we can
Okay.
avoid it.
Is that gonna be enough?
Yes, nicely done. Thank
Yeah.
you.
No snaps.
No.
That's
Yeah.
impressive.
If it's super small, you want to slurp it instead, or how do
You
you?
can slurp it.
Yeah, I think that might be easier, given how small it is.
That colony will live to see another day.
Thanks to good piloting on that one.
So
Excellent.
4 is open. Jar 4.
And Steve, is this going to be sample 29?
This is 30.
30, okay.
29 was a push core.
Gotcha.
Hey Dave, can you put bucket up on something here?
If you hit the sampling, sampling salvo, that should do it, I think.
Sampling. Okay.
That show you? Yeah, there you go.
Yeah, that'll work.
Yep.
Let's see here. We're on flush. Okay, there's 4. I guess that have to come up off the bottom here maybe. Suck up a bunch of dirt. Okay. It's I think it's on 6. Number 6. This guy. Okay, ready? Is it moving it? Do you see water moving through there? Yeah. There it went.
Nice work. A slurp sample of a Swiftia.
Not in the.
Cool.
It's not on the jar yet though.
There
There it
we
is.
go.
Okay, good. I can't see the jar camera. I'm on Herc cam. Good to know it happened just right. Could we pan left, please? Are we still pretty close to that cup coral by any chance? Looks like we've drifted off,
Yeah.
which is fine. I did get a message that
We're not that far away. If you need to, I just came up a little bit, so it's just right down to the port side. Did you want to go back down there?
I wanted to try and get one of those solitary cup corals.
Yeah.
We don't have to get that one if it's not gettable, but
Yeah, it's just right here.
okay. If we can get it, that would be wonderful. Thank you.
Hey, Bob, if
Give
you're
it a
off
try at
auto
least.
XY, I can just do a slight reset because we were sitting. Is that helpful?
Sorry. What?
Are you off auto XY?
Yeah, I'm
Okay,
off.
Great.
So we're gonna try and slurp it? Is that the plan?
Sounds
It's pretty
Sounds good.
small, so
sounds good to me.
yeah. What? Yeah, go ahead. Well, it was pretty much not. There was no dirt, you know, so we don't really need to flush it. I think it's okay. Just. So what jar are we going in?
5.
5. Okay.
So while it's hard to identify from just the visual, it does appear to be a species of cup coral that is used widely in the paleoclimate community to help reconstruct what the environment has been like over the long term. So we're delighted
Well, hang on.
to grab it with this.
We're gonna zoom in on it. Okay. Suck.
Nicely done.
It's not in the hole yet.
That's all right. It's in the tube. Let me know when it's in the hole. Beautiful dexterity, by the way, master manipulator.
Did it go? Didn't go. Where are we at on the power? 70?
Is that sample 32?
3-1.
3-1. Thank you.
Yeah, so let it off and then, yeah, turn it off. Wait a second and then turn it back on.
still waiting on confirmation of getting it in the jar, so it's holding off for a second.
Important enough to wait.
Cycle it again. Think it would have gone through there. All right.
One of the reasons that collections like this are so important for the Olympic coast is because we experience
I don't know.
fairly extreme ocean acidification and hypoxia here. In fact, the Olympic coast was recently designated a sentinel site for ocean acidification just less than a year ago. With ocean acidification, which, if you're not familiar, it's a phenomenon associated with changes in the chemistry of seawater and marked by a decline in the pH of the ocean. So it's a very important factor of ocean chemistry that helps to structure the ecosystem, for better, for worse. As we've learned over time, about 30% of the carbon dioxide released through burning of fossil fuels is actually absorbed by the ocean.
I don't know what to say about all that.
We getting the slurp in there or not so much, huh?
Well, we didn't see it go in the jar, so.
If it's stuck in the tube, right?
I mean, there's flow. It's just, I don't know, it's stuck in there somewhere. My hose is not being cooperative now. Okay, stop. Need to fix the retractor. It's not. Boy. There it goes.
Well, so Jenny, watch lead. What we'll do is we'll call that a sample. We'll try and flush it out on the surface. And if it doesn't come out, we'll just call it lost.
Sounds good, Steve. Thank you. Hopefully it won't be lost. Hopefully it's recoverable.
Yeah, I hope so. It's pretty
Are we ready
distinct.
to?
Yep.
Yeah, definitely. Are we ready to continue moving?
Yep, we can keep moving.
Beautiful. Thank you. How's the weather out there on the ship? Are you guys rocking a lot?
Pretty
No.
calm. Pretty calm right now.
Excellent. We are expecting a fairly significant storm to roll in late this evening, so we will be diving until we can't dive anymore. Is the
Hey, Dave, do
plan
you have
with
me
an
on
8
camera?
o'clock?
Could you not do that?
Oh,
Sorry.
Bob, you can go back to the dive salvo and that'll
Yeah,
put your
but
monitors
I
back.
was wondering if I was going out over the.
Nothing to be ashamed of.
Yeah, it is.
Yeah.
we move towards that thing that's right in the lasers.
The thing in the center?
Yeah.
Zoom in, Dave.
Lots of brittle stars.
This little sponge underneath it too.
Can you zoom out a little bit? I don't know where I am. All right, now you can zoom in.
Beautiful. Steve, does this look like a black coral to you?
No, this is a primnoid octocoral.
It is.
I think it's Peristanella, and it's a species we saw a few times during that transition to the Swiftia dominated. I'll let Meredith chime in if if this is something she wants to collect, but we've seen it a few times so far.
Thank you for that beautiful close-up. Love seeing the brittle stars and the little shrimp. Life begets life.
Sure, I'm
Beautiful.
sure we'll see another. So if we need to move
Yeah.
on, we can move on.
Okay. Yeah. Let's keep going. Isn't that beautiful?
Zoom all the way out, Dave.
And the time it took for that to grow to that size, even, it's pretty substantial, I would guess. I don't know how they are aged compared to some of the other corals, but some of the large corals can be decades, if not centuries old. What do you think the longest living species are, Steve, that you've seen so far?
On this dive?
On this dive or at during the seep dive?
Yeah.
You want to touch in on the zoom there, Dave?
I can speak to the corals. Obviously, the large paragorgids are pretty long-lived. We haven't seen any black corals. At least I haven't heard of any black corals seen on this dive so far. Black corals tend to be slow-growing and long-lived as well. I really not sure about the longevity of seep fauna, but tube worms also have a pretty long lifespan. I'll leave it to Andrew or whenever he comes on watch, maybe next dive, to talk more about that. But
yeah, that would be great.
I think the large paragorgids, I imagine, are probably among the oldest corals we've seen so far this dive.
Very cool. And I bet some of the sponges are potentially quite old as well.
Right. Yeah, one of the objectives, kind of along the coast, opportunistically, is to sample any fallen corals, especially bamboo corals or large primnoa colonies. Their bases can be used to age date some of the colonies, but obviously you don't want to start hacking away at a beautiful live colony to get that information. You want to sample that opportunistically.
Yeah, that's a good point. And we do try not to take the only specimen or the first one that we see, just in case they are very rare. We're pretty cautious with our sampling, particularly when we're inside national marine sanctuaries. Right now, we are outside the sanctuary, just to the west in the biogenic 2 essential fish habitat area that has been designated for protection by Pacific Fishery Management Council. Could we stop and get a zoom in on that Swiftia
one?
there? Is that an anemone beneath it? I'm
Yeah.
trying to. It is, okay. Yeah, okay, then let's keep going. Sorry. Always looking for
Bob, are you up auto
interesting
XY so I can
shots.
reset you?
Oh, you can go for it. You can see it right here.
Right, no, I just...
Interestingly, some of the rockfish can live for quite a long time, up to 130 or 140 years. We haven't seen too many rockfish in the Sebastes genus. We've seen the Sebastolobus, which are the thorny heads that seem to be fairly ubiquitous. A little deep here at about 1,200 meters for some of the species that prefer shallower depths. Is that one of the dark crinoids? What is that down below?
Zoom in,
Center,
Dave.
lower center.
Anemone.
Oh, it's anemone.
Hey, zoom out. Anemones never get the love.
No one cares.
They don't. I'm like, yeah, anemone, let's keep moving. Sorry, anemone, you're beautiful and all, but
You're not a
you know,
senator
our
enough.
time here is limited, so.
Not telling us enough.
We love you just the same. Keep doing your job.
It is frustrating too, because one of the few ways to really identify anemones is really to get them under the microscope, get them in the lab. But lately it hasn't been not a top priority. I mean, anemones are generally abundant, but they don't create habitat like corals and sponges do for other organisms. Sometimes you might find associations like polychaetes or other small crustaceans living in and around them. But yeah, they are overlooked. I agree.
Can I get a depth check, please?
We're at 1160 meters.
Thank you. We probably won't see rockfish until we hit about 600 meters, so we've got a ways to go. Thank
Yeah,
you,
up
Tom.
till the next waypoint, it's going to be pretty flat, relative. We're going along the slope, not upslope.
Looks like we have some more primnoids and Swiftia there.
You got a bit of wall here. Maybe there's more stuff.
A little hard to tell, but sometimes when you see the bright white sort of flower structures, a
octopus?
lot of
Octopus.
Oh,
times
nice.
those are, oh yeah,
Zoom in on that, Dave.
yeah, it's
Seems so exposed.
kind of a small one.
A little baby octopus. by that, I don't mean it's a juvenile. I don't think it's really possible to tell that, but small one, baby-sized. We're going to see a bunch of octopods later in our next expedition when we revisit Davidson.
That's right. They found that aggregation of brooding octopuses.
All right, and zoom out.
Bridge net.
All right, we'll quit harassing this poor guy. Move along.
Looks like I got an unwanted sample there. I'm gonna shake this off.
That's a maneuver.
Yeah.
The wet dog maneuver.
Let's see if you can get that off with Mongo. Do a
And folks
wrist
just
up.
joining
Do a
us.
wrist up and then
We are currently clearing some mud and debris off the front porch using the Mongo arm. Looking good. We're diving just off the Olympic coast of Washington, about 1,150 meters approximately, in an area known as Biogenic 2 Essential Fish Habitat. We're doing a lateral ascent up this escarpment. This nice vertical wall here, and exploring the habitats and organisms that we
Bridge.
find.
you want to move Mongo out of the way again? Get him out of the way. Is it? It's out of the way as he gets.
If possible, I'd like to set up to do another transect. If we can get the ROVs and the ship in line for that, that would be great. Sometime in the next 5-10 minutes. if
Direction we're
possible.
going?
We're going like 025.
Okay.
So I'll track a line again, but as long as we feel that you're ahead of me, or
Yeah.
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 So if you guys are good to.
Yeah.
All right. Should I mark the start, Steve?
I'll wait till watch lead is ready
Okay.
to mark the start. Let them call it.
So.
Do we have a good line and we're all set? Can we alert the ship?
Yep. I'll just start tracking the line now, and we'll just keep on a steady pace.
Fantastic. Let's throw a marker then, please.
Want to zoom in,
Thank
Dave?
you.
Oh. Do a quick zoom on this coral first then.
Okay.
This looks like a small paragorgid of some type.
What about the
Or-
fallen coral just above it? Is
What's
that
the purple
something we
thing
should
there?
sample, or is it just lying down, not fallen?
I can't tell if it's a swiftia that's just starting to
Look
die, and so
that.
its color has changed from vibrant orange pink, or if it's
Is that
something
a
really
shrimp?
different. I'm guessing it's a swiftia. That's just kind of
Oh, it's a worm.
paling, becoming more pale.
Purple guy
There
there.
are a
Oh
number
yeah,
of
it's
cup
like a
corals there, though.
platymenthes, like a
Ah,
flatworm.
nudibranch.
Oh, is
Sort
it a
of
nudibranch?
flatworm thing. Let's. Ooh, can we get a?
Oh
look
no, it
at
has
move.
Kitty.
Polychaete.
Ah.
Yeah, it's just a normal, boring. No. And this is transect 6.
Ah. All right,
Yep.
zoom out.
So yeah, that does look like a dead or dying swiftia. Tom confirmed.
All right, are we ready for the transect and consistent time and speed and all that?
Yeah, the ship's good.
Great. Let's do it. Some nice habitat. I'm going to try not to jinx us by saying things like that, but what's our altitude here?
Well, the rear of the vehicle's 5. We're on a bit of a slope, so
Okay. I'm just worried about folks being able to identify things at this distance, so
yeah, it's just
that's my question.
the front's not that high up. It's sort of a
Gotcha.
steep slope, so our altitude's taken from the rear of the vehicle.
This looks great.
Should flatten out just a slight bit in like 100 meters. I'm just going to adjust our bearing just from 025 to 030.
Steve, remind me of the stalked coral. Is it a leptos? The one in the center. Do you know what I'm talking about?
Right. What are we looking at?
White lower.
Right lower?
White.
Oh, that's
Sorry.
stalked, that single stalk coming up.
Yeah, the single stalk.
Want to zoom in, Dave?
Looks to me like a bamboo. You can just see a node, a band at the very base.
And that's why they're called bamboos. They have those like obvious nodes.
Yeah. Yeah, you can now see a few more nodes up through the colony.
It's not like other stalked species don't have those. It's just you see them on this. Is that correct? Or
No, the nodes the nodes are dark bands of protein that is unique to this group.
Gotcha.
The banding, not the protein. And the remainder of the skeleton, the white portion, is calcium carbonate.
Like a hybrid.
Usually some sort of calcite.
We're going to have to get going because we're...
Good capture. Good capture.
Yep. Let's keep going. Thank you for the close-up. We'll make sure we do a few extra minutes of transect at the end to make up for our pause here. Thank you. Meredith has chimed in that this is likely an Isidella. It's just before it has branched into its normal morphology. Thank you, Meredith. This is great video for transect, guys. Thank you. Oh, and there's an example, I think. We're not going to stop, but in the lower sort of left, sometimes you see white branched things coming out like a flower. And oftentimes, sea cucumbers can extrude their stomach to capture, filter.
Zoom in quick.
Capture organisms out of the water.
Like that one?
Looks like it.
Yeah,
that's what they look like. It's a little hard to tell from here, but yeah, that white structure, I believe, is the sea cucumber. Steve, what do you think?
No, you're absolutely right, and it's something that it's easy to misconstrue for a coral. In fact, I've done it numerous times, but I think this is Solus, P-S-O-L-U-S, or a solid sea cucumber. And yeah, you can usually tell it's a bit more pearlescent in color when you're looking at it in the light than a coral. It has this unique sheen to it. That's usually the only way you can tell unless you get real close.
Ooh, this looks like it's got nice critters growing on it. Yeah, I always look for the really bright white branching things because you figure if they're feeding structures, they're going to be cleaned pretty regularly to pull off the meals. So they tend to be quite bright and have that branching structure. So that's what I look for when I'm looking for sea cucumber species.
Yeah, they
seem to be pretty high right now.
A lot of our viewers are probably familiar with the more mobile sea cucumbers and sometimes swimming sea cucumbers, but these are firmly attached. They're sedentary for their life.
So
We're just
just a
we
heads
might,
up that at
are we,
the,
no,
we're just
go
coming
ahead.
off slope a little
Okay.
too.
Okay.
So I'm gonna bring us back up a little for after we get up.
So just a heads up that at the end of this transect, we'd like to stop and collect a Niskin, please. Looks like we're about 10 minutes into our transect. Could we do a quick check on distance, please?
Yeah, we are 90 meters right now.
Okay, good. Yep, we have a
Origin.
little ways to go. We did stop a couple of times, so
Yeah.
we'll add a few minutes. This is looking great though, ROV. Nice speed, nice distance, all the things we care about.
The distribution here looks so diverse and random. It's like smattering of all the
Yes.
things.
Well, you know, it's interesting because with this much sediment and silt, even the steep slopes tend to have a fair amount of sediment on them.
Right.
It's kind of not what you would expect. I would expect that the steep slopes are rockier, but it doesn't always turn out to be the case in the habitats here.
Right. Must play a big role in structuring the community around here.
Yeah, and I really think that the currents and the microclimates are important here as well.
It's the thing about the ocean, it's like everything matters.
That's right. That is a great talking point.
Well, it's funny because sometimes when people talk about oceanography, to me it's like, well, it's all the science just applied within the ocean. So it's not much of a subset from science, you know. But it is purpose driven, I'm sure.
Well, and it feels like there are so many questions left to ask and so many places left to visit and so many things left to study. I mean, really only in the past decade have we discovered the thousands of methane seeps that have been bubbling off this coast of the Pacific Northwest for so long. It's really impressive that even now we're still finding new habitats and species.
Yeah, for me finding
I mean,
a lot of stuff for a while.
for nerds like me, this is like, woo! And it all counts. It all matters. We're learning about, you know, the microbes and the things that are living in the sediments as well, and how important they are, not only for the ecosystem and the function of the ecosystem, but for things like the natural products that are being developed from ocean ecosystems. It's one of the reasons I was excited to work with Dr. Carrie McPhail and Andrew Thurber from OSU because they are looking at all of the microbes and the chemical compounds for natural products. Pretty interesting stuff, very cutting edge. So
Yeah.
really exciting collaboration.
What's exciting, it's like also technology enabled. So every year you have more ability to do things. So,
Absolutely. And what's great is that OET is continuing to up their game on the communications front so that we can do more and see more and view all of the extra data that's coming in from all the sensors on the ship and the vehicles. And it's Really impressive. I think we've been going about 14, 15 minutes, so let's shoot for another 5 minutes on this transect. Maybe we'll hit a few more patches of critters. We should be at about our target length by then. Maybe 5-10 minutes, we'll see.
Yeah, we're at 140 meters from the origin.
Great, thank you for the update.
Oh, ophiuroid field. Oh, whoa, a big one. Seems out of place.
Must be some pretty decent organic matter in the sediment right here because there's a ton of ophiurids.
Right.
Well, and see, so the one in the bottom center now is actually a sea star, but the
So
other ones are brittle stars.
when you have a lot of good detritus, the brittle stars tend to come out on top.
Yeah, yeah. I mean, they're deposit feeding, right? On
But versus other deposit feeders.
yeah.
Yeah. Because sometimes you just see like so many of them.
Yep. Yep.
Feasting.
Yeah.
Yeah. Sometimes it's holothuria, and sometimes you see sea cucumbers and dense aggregations around very nutritious sediment. They're basically scraping the fresh organic matter off the surface, digesting the organic components, and recycling, for lack of a better word, the waste products.
It all matters in the ocean. Our new talking point.
That concept is tough for grad students because then you feel like, oh, I should know everything. Even if you're studying a very narrow thing. Find a lot of that. A lot of interdisciplinary work.
You know, and that's one of the best things about being a research coordinator in a national marine sanctuary. I get to study and learn about so many different topics, from microbes to whales to seafloor mapping,
Yeah, that
to
sounds perfect.
meteorites. I mean, who knew? It's fun to be a generalist, but it's hard when you get the hard questions of what species is that?
Yeah.
Well,
You just have
that's
to.
why we work as a team.
Right.
Looks like we don't have a whole lot of slope in this little area. It's kind of a bit of a ledge. Is that how it looks on HYPACK?
Exactly, yeah. So it's going to be like this for a little bit. So it was steep in that last part on that, this kind of like this wave, so we're in this, depression.
Okay, well, we're at about 18 minutes. So once we hit 200 meters, could you let us know? And then we will stop transect and move